All right, so this is going to be a quick little lesson reviewing Meselson and Stahl's work on semi-conservative replication. Watson and Crick published their work describing the structure of DNA in 1953, and in that paper, perhaps in the understatement of the century, they mentioned that the structure of DNA could lead one to believe that there may be a copying mechanism associated with it. Well, it didn't take long for scientists to jump on this, and in 1958, two scientists, Meselson and Stahl, generated evidence sufficient to help us understand how this replication occurs. Before their experiment, it was postulated that replication was either semi-conservative, conservative, or dispersive. Semi-conservative replication involved the splitting of the two parent strands to form complementary daughter strands, where the new DNA molecules consisted of one original parent strand and a newly synthesized strand. And that's what you see in this example. And so the red is representing the parent strand, and then the pink is showing the newly synthesized strand. Conservative replication would have involved parent strands coming back together after the complementary strands are formed, resulting in one DNA molecule consisting of two original parent strands and one consisting of two newly synthesized strands. And that's what you see here. Dispersive replication, this was the third possibility, required that chunks of this double-stranded parental DNA would be interspersed, or kind of like a mosaic, mixed in with the double-stranded newly synthesized DNA, so that one DNA molecule would be a mixture or a mosaic of the two. And that's what you're seeing here. But it turned out that the only one that was found to be biologically significant through Meselson and Stahl's work was semi-conservative. So we're going to look at how they went about that. So Meselson and Stahl, here they are here, pictured here, they provided evidence for this semi-conservative model of DNA replication, and this was published in PNAS in 1958, and a copy of this publication is attached as a PDF to this assignment if you want to click on that and check it out later. In the interest of giving credit where it's due, it's worth noting the importance of bacteria and the availability of bacteria for this experiment. It's the combination of the simple cellular structure and their short generation time that made this study possible. And you'll see as we move through exactly what they did, how relevant that is. So Meselson and Stahl began their study by growing E. coli in a solution containing a heavy isotope of nitrogen, N15. If you can recall from chemistry, an isotope is a version of an element that contains more than the usual number of neutrons. And as a result, it has a higher atomic mass and it's more dense. And this difference in density is what made this study possible. So E. coli grown in the presence of N15 will have N15 nitrogen in the nitrogenous base portion of the circular DNA. So if you remember, E. coli are prokaryotic cells. They have the circular DNA, and after generations are copied, they're going to have to use what's available in the environment in order to form the nucleotides. And so that's why the N15 is going to be represented in that plasmid DNA. So after many generations in this media, nearly all the E. coli grown will have the N15 DNA. After several generations, a sample was collected, and these cells were lysed, and that just means broken open, in order to isolate the DNA. And the DNA was isolated and centrifuged. A centrifuge spins rapidly, so it uses centripetal force to separate a mixture based on density. And gravity would do the same thing, but it would take a whole lot longer. So this really expedites the process by using centripetal force, kind of like one of those carnival rides that you have to stand. I don't know how prevalent those are anymore, how many of you have experienced that. But you stand up and it spins around super fast, and like your face feels like it's moving backwards because you can feel everything, just the pressure. So that's an example. That's like putting you in a centrifuge. So anyway, it just speeds up what gravity would do anyway. After spinning the DNA that was isolated from the E. coli in the N15 solution, just as expected, there was one heavy N15 band. So that's what's shown here. This would be like density bands in a density column. So the heavier nitrogen is going to be expected to be here, while a light nitrogen, which is what they're going to use next, would be higher because that additional neutron makes the N15 heavier. And so after growing multiple generations in the N15 medium, they isolated the DNA from a sample of those cells, and it showed that the N15 nitrogen was what was present there. All right, so then they took some, the E. coli cells that were grown in the N15 that weren't lysed, that weren't part of that sample, were then transferred to a solution containing N14. And N14 contains less neutrons than N15, and as a result is less dense or lighter. The bacteria were allowed to divide, and the generation time for E. coli is about 20 minutes, so there had to have been a time frame here of at least 20 minutes, but less than 40 minutes, because then that would show accurate results for first generation. A sample of these cells was collected and the DNA was isolated and centrifuged, and in this case there was a single band or density layer seen halfway between where N14 and N15 bands would be. So that's what you're seeing here. Here's N14 and N15, and the band was between these two. This shows that DNA after one generation is a combination of N15 parent DNA and N14 newly synthesized DNA. So let's look back at our models. If conservative replication was the mechanism, you would have expected in this first generation to see a combination of an N14 band and an N15 band. That was not the case. So conservative replication at this point after the first generation could be eliminated. What did occur was this combination in between. That still leaves dispersive and semi-conservative as possibilities, because with semi-conservative you would have your newly synthesized, or your DNA strands would be a combination of half newly synthesized and half parental. And in the case of dispersive, you would have a combination of the two as well. So more work had to be done to determine if it was dispersive or semi-conservative, but conservative could already be eliminated. So they went on to do more. And bacteria were allowed to continue to grow in the lighter N14 nitrogen for another generation. So this is going to now be an analysis of the second generation. A sample of cells were collected, and after the DNA was isolated and centrifuged, two distinct density bands were evident in the tubes. One band at N14, lighter, matching what was in the media, and then another band in between N14 and N15. Now, if replication were dispersive, then you would expect to continue to see the band, only a single band, in between N14 and N15. So that was not the case. So after the second generation, dispersive replication was no longer viable as a model. And if semi-conservative is indeed the method of DNA replication, the researchers would expect to see the banded N14 density become more and more prevalent as the bacteria continue to divide in an N14 solution, because all the newly synthesized strands will be N14, and you're only going to see that combination of N14 and N15 in that first generation E. coli. And so they continue to allow it to divide, continue to allow the E. coli to grow, and and that was exactly what they observed. So there was a consistently increasing amount of DNA at the N14 density level, and just the same amount at the level between N14 and N15. And so that would reflect something like what's shown here. Here are these two on the end would represent your first generation grown after the E. coli was moved from the N15 to the N14, and then every generation after that is going to be N14 only. And this was sufficient to confirm that semi-conservative replication is indeed the mechanism for copying DNA.